Hello everyone, my name is Nazifa Tasnim Ahmed. My teammates are Naomi Farnaz, Alfazuddin Ahmed, Manzil Khan, and Fardin Ahmed. The topic of our presentation is how COVID-19 vaccines work, specifically protein subunit vaccines and viral vector vaccines. First, let's take a look at the current COVID-19 situation around the globe. The first appearance of the virus was noted on December 1st, 2019. The number of the countries that have been affected by the virus is 221. The number of the cases of infection have been 114,689,260 and the cases of fatality have been 2,543,310. Vaccines are one of the most impactful ways to prevent COVID-19 infection. Till now, 55 vaccines have been developed. Vaccines are developed through research, production, and trial phases in accordance to FDA guidelines. Currently, 4 types of vaccines are being administered throughout the world. They are inactivated vaccines, which are non-pathological, mRNA vaccines, protein subunit vaccines with spike proteins, and viral vector vaccines.
Rather than conventionally using the whole virus cell, the protein spikes are used to trigger immune response. These protein subunits cannot infect the host cells and are incapable of causing diseases. However, they trigger weaker immune responses due to their inactivity and require booster shots and adjuvant antigens for stimulating improved response. Viral vector vaccines achieve a strong immune response by inserting a small fragment of genetic code of the pathogen into a host cell, in this case, the SARS-CoV-2 virus. Unlike the mRNA viruses where the genetic code is inserted directly into the human cells, harmless virus is used which effectively acts as a delivery system to get the code into the cell without causing a disease. By hijacking the cellular function, these vaccines mimic the way viruses normally reproduce during natural infection, but rather producing active viruses, the patient cells produce large amount of antigen. This triggers a strong immune response without booster shots or adjuvant antigens.
As we know, the protein subunit vaccine contains purified pieces of a pathogen rather than the whole pathogen to trigger an immune response. So the main advantage of protein subunit vaccination is that it is a well-established technology that's advantageous for those with compromised immune system. This type of vaccine requires no live components, so no risk of vaccines triggering disease. Also, this type of vaccinations is relatively stable. On the other hand, viral vector vaccine introduces a safe modified version of the virus known as the vector to deliver genetic code for the antigen. So the viral vector vaccination is another well-established technology that can trigger a strong immune response and is also involves both B cells and T cells. This type of vaccine shows a highly specific gene delivery into the host cell with a vigorous immune response. It avoids handling of any infectious particle and it has been used widely for SARS-CoV with positive results from the trials. The main challenge for protein subunit and viral vector vaccines is that this type of vaccines are relatively complex to manufacture compared to others. For protein subunit vaccines, adjuvants and booster shots are required. Also, determining the best antigen combination takes time. For viral vector vaccines, the host may possess immunity against the vector, which reduces its effectiveness. This also may lead to cancer due to integration of viral genome into the host genome. The side effects of these two COVID-19 vaccines are similar to those of other vaccines. Most are mild to moderate and occur within the 3 days of vaccinations. Pain and soreness at the injection site is the most common side effect. Other normal potential side effects are fatigue, headache, chills, fever. The following side effects have been reported: muscle and joint pain, delayed swelling, redness and rash at the injection site, swollen lymph nodes, especially manifests as a lump. Most COVID-19 side effects can be treated safely at home without any sort of special care. Over-the-counter pain medication like ibuprofen or acetaminophen can be used to reduce pain or discomfort at the injection site. Drinking plenty of fluids and dressing lightly can reduce discomfort from fever.
Universities have been working with protein subunit vaccines. Novavax, University of Pittsburgh, Premas Biotech, India, University of Queensland in collaboration with GlaxoSmithKline. The testing process consists of 3 phases. Phase 1 trials focus on the intensity of immune response. Phase 2 trials focus on dosage and delivery schedule. Phase 3 determines safety and efficacy among a large primary use population of the vaccine. Both the trials mentioned in this presentation are Phase 3 trials. We are now going to look at the clinical trials conducted by Novavax. There were in total 15,000 participants aged between 18 to 84 years old. Among them, age of 27% participants was above 65. Novavax picked participants from crowded conditions working in close contact with public because they had greater risk of infection. The participants were then divided into 2 groups. One group was given the COVID-19 vaccine and the other group was given placebo injection. The results were praiseworthy as the overall efficacy came out very good. Participants were tested for the presence of antibodies. Efficacy by strain was calculated to be 95.6% against the original COVID-19 strain and 85.6% against the inland variant strain.
The following mentioned institutes are the most notable ones producing the viral vector COVID-19 vaccines: Oxford University, Johnson & Johnson, Gamaleya University, and Serum Institute of India. To understand the efficacy of the viral vector vaccines, we are now going to look at the procedure of the clinical trials conducted by the Oxford University and also observe their results. In the clinical trial, there were a grand total of 11,636 patients between the age group of 18 to 55. The participants were then divided into 2 groups. One group was given the COVID-19 vaccine, the other group was given a control injection. Observing the results published by the Oxford University, it was seen that there were no hospital admissions in the vaccine group, and only 175 severe adverse events were reported. These adverse events included transverse myelitis, hemolytic anemia, and fever higher than 40 degrees Celsius. Now let us look at the efficacy calculations. To analyze the efficacy of the vaccine, the participants were first screened by testing for the presence of antibodies following the first dose. After the second dose, the efficacy analysis was conducted, which involved observing COVID-19 occurrence 14 days after the second dose. This process was continued for a period of 4 months, and during these few months, it was seen that 70.4% of the participants did not show any COVID-19 symptoms and was not tested COVID-19 positive either. With the pandemic completely disrupting our normal lives, it has become absolutely necessary to manufacture as many possible variations of the COVID-19 vaccine. By making use of appropriate manufacturing process and appropriate clinical trials followed by efficient vaccine rollout programs, it will be possible to beat the pandemic and go back to our normal lives. Thank you.